We're here at the Institute for Genomic Biology on the University of Illinois campus to talk about a topic that probably few people have heard about, synthetic biology. We're going to meet with a group of students who are preparing for this year's iGEM competition. We're going to talk about synthetic biology and what it might mean to you on this edition of Surrounded by Science.
Hey, Allison and Dylan, what if I told you you could get a head start on college before you graduated high school and for little to no cost?
Well, I'd ask you what the catch is.
Yeah, how can I take college courses if I haven't even finished high school yet?
That's easy. You simply enroll in dual credit courses at Parkland College.
But won't that be expensive?
Yeah, will all the credits transfer?
Well, most four-year universities accept these credits, and you're actually saving money. Many times the high school will even cover the cost of the course.
I'm sorry, did I overhear you guys correctly? Did you just say that you could take college courses without paying college tuition?
Yeah, all we pay for is the course and the textbook, and we can earn college credit towards getting our degree.
You guys are catching on. The credit works toward a 2-year degree, a 4-year degree, and even toward career certificate programs.
I like the sound of this.
I don't know, this all sounds great, but I already have a busy schedule and I won't have time to drive to classes.
You won't have to. Parkland offers a number of dual credit courses online in addition to the on-campus courses. Some of the courses are even taught in your high school.
You mean to say that one of the high school classes I'm taking right now could work as a dual credit course as well?
Yes, you should talk to the school counselor to see which classes are offered as dual credit in our school. Then all you need to do is apply to Parkland, and once you're accepted, fill out the application to the dual credit program. You'll need the signature of your parents and advisor for this.
That sounds easy enough, but are there any requirements to being eligible for dual credit?
Yes, you must be a junior or senior in high school, age 16 or above, in order to be eligible for dual credit courses. You can print an application online or visit Parkland's admissions office to pick one up.
Well, I don't know about you guys, but I'm going to see the counselor right now.
Yeah, I don't want to miss out on this opportunity.
Me neither.
Get the ball rolling to a new career with an Eastern Illinois University degree at Parkland College.
Parkland and Eastern really made it work for me.
Now I'm taking Eastern classes on Parkland's campus. I'm working on my bachelor's degree from Eastern without commuting.
Thanks to Parkland and Eastern, I'm reaching my goals.
For more on Eastern at Parkland, call 217-351-2543.
Okay, we're here with Colin, who's part of one of the 2 iGEM teams, and welcome. Thanks for being on the show.
Yeah, thank you.
And which of the 2 teams are you on?
I am on the software side of the U of I iGEM team.
Okay, and why is there a software side? What sorts of things do you do on the team?
So the software side, it really deals with the computational side of synthetic biology. The wet lab team works in the lab with cells and bacteria, whereas we on the software team, we code and we build software that allows other synthetic biologists to more easily design their work and helps them do stuff in the lab. So
the 2 teams work together pretty closely?
Yeah, well, actually, we are currently helping the wet lab team model one of their cells that they're working on, and this entire summer we actually had joint meetings, and you know we really were working side by side, separate projects, but we you know talked, we shared ideas with each other.
Now, as a physics teacher, I don't know very much about synthetic biology, but how would you describe it, just maybe from a computational standpoint? Like, what is synthetic biology?
So, synthetic biology is really a compartmentalization of biology. A lot of people see biology and know biology as it's not very standardized, and so one of the biggest things of synthetic biology is make it standardized so that it's easy to engineer. And with the computation side, we take the standardization and we can use it to build software programs to make some of the design processes a lot more automatic and easy to do.
So you're making, say, is it fair to say you're making models?
Yes. Actually, our current, the project we're working on called Biomortar, it has 3 parts, and one of them is a modeling program that allows you to model a theoretical cell that, that the program generates.
Anything I haven't asked you that I should ask you?
So, our current project, Biomortar, it has it's a 3-part genetic design suite that that automates the design process for designing metabolic pathways into bacteria.
Interesting. And all with software that your team has developed?
Yes, actually. Most of it is actually coded for in Python, which is an open source development programming language, and the user interface was coded for in it's called Django, and it's a web development programming language, and it really allows for a very easy user interface, as well as the ability to host it on a website very easily, which gives greater access to it. And then finally, the cell modeling is coded for in it's called the Cobra Toolbox. It's a toolbox for MATLAB that does a lot of helps with a lot of the calculations that need to be run with that.
I'm curious, what year in school are you?
I'm actually currently a sophomore in bioengineering.
Oh, and I was going to ask your major too, bioengineering. Okay.
Yep.
And where do you hope to go with that?
So I'm hoping to maybe go to graduate school, or maybe take it into use it for starting up my own business. I'm not too sure right now. It's only sophomore year.
That's okay.
But yeah, okay. So it should be pretty exciting. Yeah.
Okay, well, great experience, and thank you very much. We're here with Stephen in the iGym lab, and tell us a little bit about what team you're on.
I am on the BioWare team. What we do a little bit is actually work physically with the DNA so we can submit it as into the competition.
And what sorts of procedures do you work on here in the iGym lab?
A few procedures that we do, since we do physically work with DNA, we do DNA extraction, is where we actually grow out the bacterial cells, and then using a series of reagents, we extract the DNA and then engineer it using other particular enzymes known as restriction enzymes.
Okay. And how is this a part of this whole competition that you'll be participating in soon?
Well, the competition is a synthetic biology competition, and essentially what synthetic biology is an abstraction of genetic engineering. And really what that means is making genetic engineering more accessible and easier to other lab technicians and the sort. Some of the procedures that we follow in order to produce these genetically engineered bacteria is to, as I mentioned before, extract the DNA, and then after that we manipulate the genes, whatever genes we in particularly want to use, and we insert them using another various series of protocols in order to put them into the bacteria that we'd want.
And where do you expect to go with this? Do you expect to make a career out of this?
Hopefully I'm expecting to make a career out of this, yeah. Currently I'm trying to apply to graduate schools for environmental engineering, and one of the areas that we're trying to compete in right now is actually in an environmental aspect with our bioremediation project, where we're actually trying to engineer bacteria to alleviate arsenic out of water.
We're here with Megan, who's the director of the BioWare team. Thank you for being with us today.
Yeah, absolutely.
Why don't you tell us, just start off by what is iGym?
So iGym is a really great opportunity for undergraduate students. The idea is to take a synthetic biology project. So you get together a group of undergraduate students, starting in the spring usually, and at that point you come up with a project and you do a lot of the research behind it. And then we do a lot of fundraising and find a lab space, so we stay on campus over the summer and actually complete the project. So that's a lot of interesting lab work and a lot of time that we spend learning. And then in the fall, in November, we go to MIT and actually present about all the research that we've done so far.
How are the students chosen to be in iGym?
That varies a lot based on the team. For the U of I team, we have an online application that they fill out, and then we do brief interviews. And it's usually the past team members who really organize the whole organization, and they're the ones conducting the interviews and choosing the next year's team.
What would you say the goals of the program are? What do students really want to get out of it, besides a medal at the competition?
Yeah, no, a medal is a great part of it, but also we really enjoy the lab experience. Is a great thing to go on and work in other labs. It really helps if you're going to grad school, but also it's a really fun experience to get to work with other students your own age on a project that you come up with over the summer, and a lot of them apply strongly to real life. For example, Berkeley did a backdoor blood project, and the ultimate goal there was to create a system that would be able to survive in your bloodstream, so one day they could use it to deliver drugs.
Interesting. What is synthetic biology?
That's a million-dollar question. So synthetic biology is the application of engineering principles to biology. And for engineering, some of the big things that we deal with are abstraction and standardization. So if we can take biology and break it up into parts and have standards and protocols that work on a regular basis, and then use those different parts and be able to put them together in new ways, that's when we can start engineering the system, and that's when you get into synthetic biology.
And how is this different from, say, genetic engineering? Is it close?
It's definitely a fine line between the two. For genetic engineering, it has to do more with restructuring the protein itself, and on a very more specific, working on like different nucleotides on a lower level. And then synthetic biology requires more abstraction, so you work with parts and you pull those parts together in new ways to create a new system.
What sorts of organisms are used in synthetic biology?
Mainly we use yeast and bacteria. And the best type of bacteria to work with is E. coli. So a lot of times people aren't familiar with that, and you hear bacteria and you think it's something negative. But all the bacteria and cells that we work with are non-pathogenic. So they've been tailored specifically for lab work, and they're not harmful to humans.
Are any ethical issues in working with something like this, or?
We are in a biosafety level 2 lab, so all of the students on the team have taken 2 different exams through the biosafety division at the U of I. And those are basically to show that we understand the lab concerns and we will conduct ourselves safely in the lab. So there are several concerns in working with synthetic biology and DNA, but they are primarily addressed by that, and we also have an advisor who's careful about the requirements that go along with that.
Since we're going to be broadcasting this to the general public, why should somebody sitting in their easy chair out there be concerned with synthetic biology? Like, why should they care about it?
Well, it's really a new field, and synthetic biology is something that's very interesting for me, and it helps that I've seen all the applications for it. So one specific example that helps me realize why it's important is the work that Jake Kiesling from Berkeley has done in finding a cure for malaria. So he has taken the genes from a plant called artemisinin and transferred those into bacteria. So now the bacteria can produce the cure instead of having to go all the way to the plant to get it. So this will hopefully make it a lot cheaper and a lot more accessible to the public.
So you maybe see possibly some medicines coming out of something, the research?
Definitely.
Very good. Are there any environmental issues that synthetic biology can help out with?
One thing in particular that our project deals with is water contamination. So it's somewhat of a detailed project, but the idea is to use bacterial cells to collect arsenic that contaminates a lot of drinking water. And we'll use the cells to collect the arsenic, and then we'll collect the cells themselves, and that will take care of bringing the arsenic out of the drinking water.
Is this something you'd like to go into as a career?
I think at this point I'm heading towards grad school after college, definitely. And it's been something I've been interested in since hearing about it as a freshman here on campus.
How many years has the University of Illinois participated in the iGym competition?
This is the 3rd year, and I think we've improved a lot each year. It helps to have the past experience.
Very good. Were you a part of the last year's winning team?
Yes, I was part of the last year's team, and then this year I've been director, so it's been great to work with the team again and see it improve on a yearly basis.
Roughly how many people? I know there are 2 teams, is that correct?
Yes.
What do the 2 teams do? What's the difference between them, and how many people are on these teams?
Well, it's varied a little bit from year to year. This year we have 12 people total, and there are 8 people on the BioWare team and then 4 people on the software tools team. So the BioWare team is what you've probably heard most about today, and that is what I'm part of. So we're doing something actually in the lab coats, working with cells and DNA. So the software tools team is a different idea, and they work with designing some sort of software in order to come up with a program that's useful for synthetic biologists. So I think you talked to Colin about their work, and basically some sort of program that can organize all the data that synthetic biologists collect or manipulate it and help us out in that way.
Now, what haven't I asked you that you want to talk about? And it may not be anything, but I want to give you the chance.
Well, I'm happy to talk about our project specifically this year. As a general idea, we've worked with logic gates in bacteria, and we've also worked with the bioremediation part that I mentioned in collecting arsenic. So going back to the logic gates, that idea is where you have some system in a cell that looks at what's coming in and decides how to regulate a certain gene. So a certain gene is either turned on or off based on an input. So that's like the idea behind a logic gate. And we've been working with those and making more complex logic gates, and this will allow us to be able to control a cell a lot more closely. And by being able to control cells more closely, we can manipulate what they do and have them produce the right kind of medicine or other things like that.
Okay. Best of luck this year.
Thank you.
In the mid-70s, law enforcement was not really ready for women. It was tenacity, it was determination, and support from Jim Whetstone that kept me focused. Parkland enabled me to acquire a bachelor's, a master's, and I am a graduate of the FBI National Academy. It was more than just criminal justice. Parkland gave me a true education.
As an instructor, I view academic honesty as very important in the classroom. I want the students to not be deprived of their learning and learning how to learn material. And I view that if they look at someone else's paper or try to get information from someone else without thinking about it, then they're depriving themselves of the learning process.
It's not just about covering bases. It's about making sure that you're conducting yourself in a very professional fashion. In my opinion, personally, that's always the thing that wins the day, is doing what's right, even when nobody else is looking.
I want to be truthful in what knowledge I know and sort of promote a role model for my students.
Honesty not only serves the college very well, it serves the student very well.
Honesty and integrity.
Honesty and integrity, Parkland College.
Parkland College.
We're here in the biology lab with Mr. Scott Seekin. Good morning, Scott.
Good morning, Dave. How are you?
Very good, thank you very much. And what courses do you teach here at Parkland?
I teach a couple of courses here. I teach Biology 101, the non-majors biology course, and I also teach a couple of sections of 141, the Biology Majors Introduction to Biology course.
And you had the student team from the university come and visit your classroom.
I sure did, and boy, was that an interesting experience for my students. they came to talk to us about their project, their research project they're doing at the university on synthetic biology.
Yeah, and now in your mind, what is synthetic biology? I know we asked you by people that, but...
Yeah, that's a great question. Synthetic biology is kind of a new field just in the last, well, 5 years or so now, that involves just what the name implies, synthetic, building from scratch biology. So we're taking materials we find in the living world and putting them together in new ways that we essentially design and think will benefit ourselves as human beings or the world in general to support life. And so we are truly building life, in some cases almost essentially building life forms and rearranging life forms that are out there in ways that will benefit people.
Just look in the crystal ball for a second. What do you see the implications of this? What could potentially come from it?
Huge implications for the future. Designing, one thing that life is very good at is reproducing itself and taking on characteristics. And if we can get it to, by manipulating it at the smallest level, if we can get it to take on characteristics to do things that will essentially benefit us, this could change our world dramatically for the better in terms of helping us detect and remove pollution, detect and remove toxins, detect and remove diseases within a body, just all sorts of possibilities for where this type of thinking and this type of engineering can take us.
It's interesting that the team came and visited a non-majors class. What do you think your students got from the presentation?
That's great. I think they got a lot out of the presentation because, as you say, they are non-majors. They are people who don't plan to spend their life in biology and yet spent a whole semester with me learning as much as they possibly could in one semester about how biology works. And so they got several things from this interaction. First of all, they got to see how some of the ideas that we talked about in class get put into application in the real world of research, and how those fundamentals that we talked about all semester long are actually being used to do new and interesting things in the world. So that's one thing. Another thing is just reinforcement of the ideas. I mean, it was close to their finals time, so they were trying to remember as many of the concepts from all semester. And so this was a great way for them to get a quick review of all the fundamental, really important parts of biology that we've been talking about all semester. And then to see what their peers who are biology majors and do plan to spend the rest of their life doing something with biology are actually what they're attaining while they're still undergraduates, and seeing that they are accomplishing new and interesting things in the world of research as undergraduates, of course under the guidance of some professors. So one final thing I think they got from it was an understanding, a broader understanding of the careers that are available with a degree in biology, because there is a wide range. It's not just research. Of course, there's also teaching, there's also writing, there's also just a whole variety of possible careers that are available to people who take biology as their major course in college.
And I think it's great that there is this collaboration. Could you comment just on the collaboration, the idea that people from the university are coming here to Parkland College? Is that a good thing to have that university-Parkland collaboration?
I think it's a fabulous thing. The university is a real resource in terms of the researchers and the ideas that are going on, and Parkland is benefiting hugely from that interaction, and it's benefiting our students directly, and it's keeping Parkland at the front edge of understanding what's going on in the world of research and how we are moving the scientific process into the future. And to have direct access for our students to that, I think, is something that really sets Parkland apart and really makes Parkland a premier institution.
Hey, Scott Seekin, thank you so much for being on.
You're most welcome, Dave. Thank
All
you.
Right, thank you. And thank you for joining us on Surrounded by Science. So come here often?